Thank you very much, Julie, for the opportunity. And hello, everyone. I am Nalinava Sengupta, born and raised in the city of Joy, Calcutta, and then moved to Indian Institute of Technology, Mumbai, to pursue my PhD. And during those days, one of my closest cousin sisters suffers cancer. And I accompany her always whenever she had the treatment. The good news is she survives till dead and healthy. But during those hospital visits, she had to take surgery, radiation therapy, and chemo. And the chemo part was toughest. And she lost appetite. She started feeling numbness in the leg and whatnot. And experiencing those scenes in the Indian hospital, I see 70 other chemo patients are also going through similar journey. And my PhD in organometallic chemistry was indirectly related to cancer research. But during those hospital visits, I start thinking, if I am not trying to solve the real, genuine problem, what am I going to do by publishing in reputed journals? And then I decide that I'll be pursuing on cancer research to develop better cancer drugs as a young chemist, young organometallic inorganic chemist. And then I was fortunate, A, that she is healthy, fit, and fine. And then B, I get hired to University of Cologne, Germany, to pursue my postdoc exactly on synthesizing better cancer drugs based on cisplatin. Maybe he knows very well. Now, I was doing very well during those and published in Organometallics and Journal of Organometallics. And meanwhile, my family started seeing good match for me to get married. And then I try to move for more salaried postdocs, and I moved to Norway, Sintef. And then my wife, she was a master's in molecular biology, and she joined. So I'm lucky on third count that she joins as a molecular biologist, starting a PhD in Oslo as well. And her project was why modern cancer drugs are getting resistant, exactly on DNA repair and mismatch and repair field. And then she studies about a popular drug named 5-fluorouracil. And she finds that this should not be given anymore because it's no more same efficacious. It is resistant because the oncogenes have become smarter over 15, 20 years. And now oncogenes understand that this molecule is going to kill me, so I mute it. So it became like my observation that good publications are only up to better citation. And after that, nobody is taking care. So it happened like after her PhD, our purposes match and our brains couple. And we test out our idea to validate it with professors in the University of Oslo. However, professors praise it as well, and my wife's lady supervisor also joins as co-founder. And the Research Council of Norway says, if you form a private capital stock company, then you apply for funds, we may grant you the research money. But then we keep on trying, and we really got 200,000 kroner, which is like 20,000 euro or Swiss franc, to start as a pre-project. This one, right? Yeah. Oh, I go back. Which one is? Oh, yeah. So we come up with AI-backed, targeted, and rapid cancer drug delivery system. And it should be innovative nano drug. So that's why we name it iNANOD. And we do see that there is an opportunity to create an integrated system where his lecture, you see, there were three branches. But we do believe now these three can also be combined into an integrated system where a nanotechnology or nanoparticle, through its enhanced permeable and retention effect, takes passive targeting to the cancer site. But however, the statistics of small molecular drugs and nanoparticle are very poor when you calculate with respect to the dose injected. So the which button is next, please. Yeah. Yeah. So we do believe that through nanotechnology, we will be enabled to generate chemotherapy to kill the cancer tumor from inside, and exactly what he was saying, to generate CAR T cells from the outside. And we do know about special types of groups of molecules which can actually penetrate the tumor, so that the target specific and chemotherapy and immunotherapy will be faster, so that the exponentially growing tumor doesn't win the battle against the therapy. So that's the whole idea. Because when one chemo given, for the next chemo, since the small molecule spreads all over the body and it takes 3 weeks, 4 weeks time to get rid of that excreted from the body. So next chemo session becomes prolonged and longer, and cancer patients rather suffer from side effects, and probably sometimes cancer wins. So to deliver on the promise, we assembled exactly experienced people together with us. I introduced myself, of course, and Professor Hilde was my wife Tanima's professor, and they are molecular biologists from neurobiology to oncology, everything they do on DNA level. Åge Nardal is a trained pharmacist and was country manager of GSK for 13 years, and he also running a clinical trial for insulin through skin. Per Foss retired as a Director General of Norwegian Patent Office and then joined our board and helps us to strategize on our patent strategy and policy, etc. Thomas is present here, and he helps entrepreneurs to get funded. And it's like nearly 27 years he is working for entrepreneurs. And the great news is he had been to a couple of FDA meetings. So when I introduced ourselves to Thomas, he says, well, you have done PhD, but it will be a thorough grilling for you if you really reach the FDA meeting. And Steinar Queensland is most probably the best oncologist Norway has ever produced, and he visited that Indian hospital 7 times in his lifetime for research work and conferences, and was Corona Commission 1's chairperson of Norway. Next, please. And we do have advisors, and one of the front runner is Knut Smerud, who runs clinical trial research organization from Norway. And Espen is another oncologist to provide us a second opinion. And Bruno is from Switzerland, who helps us to grow here as well. And Ketil, Gokche, they help me to on legal aspects, etc. And Jorn is the first angel whom we met, and he raised a fund, friends, family, and fans round. And that's how we could raise Norwegian 3.5 million and could survive the COVID, etc. Next slide, please. So that's the statistics. Even though you have torpedo or even though you have immunotherapy separately, the real challenge in my eyes, or many scientists or say core chemists, we see it like kind of a failure when the median statistics is 0.2%. And when a small molecular drug commonly used named paclitaxel was given to a mouse, And then again packed in a nano formulation, then it was found 0.2% reached the tumor inside when it is a naked drug, and when it's packed as a nano formulation, then 0.6%. So passive targeting made 3 times more drug deposition. However, it's not that great. Next slide, please. So these are the side effects, and I am not going to explain it. Now we know all of them. So real challenge is the low medicine uptake. And here, our platform technology can really do something, we believe. Next slide, please. So these are like penetration and the faster delivery. And of course, we would like to have monoclonal antibody added. And we don't play with innovative drugs. We would rather try a tried, proven, tested drug inside to come up with first proof of principle. Next slide, please. So our focus is, we will name it. It's still not registered, but there is a peer pressure that we name it Nalinava. And that will be our platform technology to generate that integrated system where you can actually deliver drugs decisively, target specifically in a faster way. Next, please. So we made a prophetic patent, and we do believe we will be able to increase the statistics 75 times more, because the way that special group of molecules, they actually worked on mice on a pancreatic cancer, which is a very difficult cancer, and made a great drug deposition. And when I was reading that article, I was initially very nervous. Probably my startup ends there. When in the middle, then I study, and I find, no, they are not targeting exactly like us, and we do have other multi-steps in our sleeves as well, so our prophetic patent survives. But at the same time, those scientists who were criticizing, probably you are banking too much on those special molecules, they are silent now. So that molecule has shown that it has got the real purpose and real pace to deliver the drug. So we select liposome as the safest nano vehicle, and then we will be anchoring monoclonal antibody, and we will be using polyethylene glycols to graft them. And also we will be grafting our special molecule so that it generates a multi-step drug delivery system. And there will be a little bit of engineering on the polyethylene glycol as well, so that the linkers do their job in different conditions differently. And therefore, we believe that up to 30% of the injected dose can be delivered. Of course, the AI will be thoroughly implemented. And that's why it's still postulate, one can say, but we carried out our first study through computational biology to mitigate the risk. And I did that with one of my batchmates in masters. He is running computational biology service company from Bengaluru. He is brutally honest. Had I selected bad things, he would have told my wife even. So, and criticized us. But the good thing is he came up with the comment that, oh my God, looks like we think it should go to nature or science. You have designed it so well, and the concentration ranges and all in the reported we select is such that there is a huge chance that the injected dose will remain stable till it reaches the target. So that much R&D development we could do, and we never could raise $1 million or 10 million NOS kroner to actually try it on mouse. So we are fundraising. We are also crowdfunding at the moment. So still there is a place to fulfill that gap, and we can start tomorrow if I get an investment of that size today. So wait a minute. No worries. So the as you see, that's called immunoliposomes with monoclonal antibody grafted, and we will be using oxaliplatin because it's more aqueous soluble, so that it goes into cytoplasm and takes no time to hit the DNA and make the apoptotic pathway. There is a similar formulation, immunoliposome, actually from Switzerland, was tried against triple negative breast cancer.
It's our former company.
Oh, yeah. But we do believe it failed because it lacked the tool we have.
And then you need to tell me what you have.
Yeah, then you need to sign an NDA. Yes, please. Next slide, please. Yeah. That's the 0.2% for over 30, 40 years statistics, and this is like over 15 years statistics. And when you do active targeting with monoclonal antibody or any enzyme which can target the tumor microenvironment, then the statistics becomes 3.5%, median statistics. And we do believe that 3-4 times greater it will be when we use that special molecule or special group of molecules for that effect. And then if we, from up to this, it's platform technology, but then AI takes it personalization. So then we feel there are 120 types and subtypes of cancer, and 90% of that is solid malignant tumor. So like 100 types of cancer tumors, we will be able to address if we get solid support through AI on mapping the tumor and fine-tuning the formulation. So we dream a day when the doctor will diagnose the patient and tell the pharmacist or chemist or druggist to say, okay, this patient has this particularity in the disease. So we do this amount recipe A with A is to B is to C is to D, this ratio. That will be perfect so that there is no side effect and the cancer is shrinking through the double effect of chemotherapy and immunotherapy together. Next slide, please. So that's why we strongly need AI support, of course, to tumor profiling and map selection, then determine and guide optimum surface ratio between these, and determine the chemical composition and overall stability, and finally that rapidly discover that personalized best fit. Next slide, please. So these are the market slides, and these are based on predictions, of course, and this market is growing, and also nanotechnology. After COVID, big pharma is also grabbing this lipid nanoparticles and liposomes and AI companies, which can actually work together and usher those days faster. So we do have a competition, and we do feel that if we don't do it, somebody is gonna do this. Next, please. And these are, of course, our platform technology, but first you have to prove it on at least one cancer. So we select epithelial ovarian cancer, and we do perform that AI stuff, and that's the market. And due to lifestyle, modern lifestyle, women after menopause are more exposed to epithelial or ovarian cancer, and it gets diagnosed on phase 2 or 3. The first stage, it's silent killer. So the market is gonna very much up, so it's very relevant to develop a good drug against epithelial ovarian cancer, and current success rate is 47%. That means this is neither a very difficult cancer nor a cancer which we say we can cure it. No, there is still a lot of space to cure it. Next, please. This is secondary target market, of course, if we target all other cancer types. Next slide, please. Yes, what is our USP is not only active targeting, but this special penetration. So others are happy to target, and still that is a great achievement, of course, but we think we should be able to push the boundary with the pace as well. Next, please. So we have supporters. We are member of Oslo Cancer Cluster, and then Norway's Venture Lab and the Life Science Cluster as well. And we got support from Research Council on taxation, etc., and small grants. And Innovation Norway supported us with mentoring support, etc. And Invent2 is a tech transfer company with University of Oslo, but we have a typical special, not typical, rather special agreement with them for one typical case with them. And our model is we don't invest on recruiting too many people or building on infrastructure. Rather, I find there are many great service providers for R&Ds. So we make good contract with them and we will enrich our patent stage by stage. Next, please. So this is typically for investors. Companies like us are not available for invest usually. So we are pretty unique because mostly it happens through push mechanism of innovation where the laboratory and professor thinks for 6 months whether this has merit for further innovation, then it's given to TTOs, and TTOs think for 3 more months whether they have enough investor network and muscle strength to push it on the market. But here, with 3 scientists coupled in our brains and solving an unmet need, and the market is pulling us for the innovation. So first option for an exit is when we have the proof of principle after mouse studies, and then we approach VC funds, or we try to hook up with co-development with a big pharma partner on upfront model and milestone-based payment, and then royalty-based payment after exit 2. That's co-developmental model. So these are, if we reach there with good or great quality data, we are sure there are many pathways to flourish and expand so that we don't confine ourselves only on epithelial cancer drug, but also say head and neck cancer or non-small lung cancer or etc. over other cancers as well. But this slide and calculations are for one single product so far we calculated, please. So now we are raising funds, $1 million plus little, but we raised so far 0.4. So there is space for $1 million still. And we value ourselves $4 million US. So it's like 25-26% post-money dilution, and 90% is R&D, and I take a salary like a PhD student. And we believe we'll be able to deliver the POP by 2026 quarter first. Next, please. There is a video, I don't know whether it can run. We can skip that. We are on Folke Invest, a Norwegian prime platform, and if you're interested, I can share that link through LinkedIn or anywhere. Yeah, I can share. You can share, please. But the language is in Norwegian. You got to change it into English, and we are working on an English page as well. Next, please. It's a disclaimer that you take risk in startup when you invest. And I wake up at 5 o'clock to usher that day that you find that your next day neighbor or close relative had cancer, got treated, but there is no sign on the body or face. Thank you very much for your attention, and I'm open to questions.